Hi, my name is Maya, and today we're going to be talking about enzymes. So imagine that you're at a party and there's a stranger on the other side. Usually you wouldn't interact, but a mutual friend can get you guys to interact. In the same way, enzymes are biological molecules, usually proteins, which bring together molecules and causes them to react. They catalyze these reactions, meaning they speed them up without being consumed. But how? This has to do with the transfer of energy. Without enzymes, there are three ways that chemical reactions occur. There's actually an acronym for this called CEO. C is for collision, and then energy and orientation. When molecules collide, they react. The second way is energy. By adding thermal energy to a system, the kinetic energy is raised, meaning that there's more collisions, because as there's more movement, there's a greater chance of a collision and thus a greater chance of a reaction. Finally, the molecules need to be in the correct orientation to react. Think of it as a puzzle piece. They can't just go together in any way. They need to be correctly oriented in order to fit in together. This process is made a lot easier through enzymes, which lower the reaction's activation energy, which is the energy required to initiate a chemical reaction. If we look at it graphically, the amount of energy needed for a reaction to occur with enzymes is drastically lower than without enzymes. So these enzymes, by bringing molecules together, are actually lowering the activation energy needed for the chemical reactions to occur. Now, we're going to talk about the structure and shape of enzymes and how that determines their function. Enzymes start off as long chains of amino acids, which is primary structure, and then they are folded into secondary structure, tertiary structure, and quaternary structure. And the majority of them stay in quaternary structure, which is when they're all folded up together. Most enzymes have a globular shape, which means it's in the shape kind of as a circle, if you see it here, with pockets. And these pockets are the active sites where substrates go. The substrates, which is another word for reactants, fit into the enzymes to then react, and this creates an enzyme-substrate complex, the combined structure of an enzyme with a substrate. The enzyme needs to fit into the substrate. In this case, it wouldn't fit. Specific enzymes react with specific molecules. This can be seen with lipase and lipids or ribonuclease and RNA. And there are two types of enzyme models. There's the lock and key model. In this model, each enzyme has a specific active site, which is where the substrate goes, that matches to a specific substrate. In this case, think of it like a key. You need a certain key in the correct orientation to work a lock. In the same way, the substrate needs to fit perfectly into the enzyme to create the products. The other model is the induced fit model. In this case, the substrates and the enzymes don't initially seem to fit. However, the enzyme changes its shape to accommodate the substrates, and so the active site is altered, and so a product can be made. In both of these cases, whether it's the lock and key model or the induced fit model, the shape of both the enzymes and the substrates is what allows them to react and function. In some cases, in order to bind to the molecules, some enzymes need coenzymes in order to react and connect with the substrates. Coenzymes are inorganic molecules, and some examples could be potassium, iron, or zinc. Take this case, for example. This enzyme has a very specific active site, and not all substrates will be able to fit in. So, a coenzyme that looks like this can be placed into the active site so that a substrate that looks like this can fit in. Now, let's talk about feedback control mechanisms. Enzymes use lots of these in order to regulate how many products they create. This starts with allosteric regulation, which is the regulation of enzyme activity using activators and inhibitors, which bind to the allosteric sites of an enzyme. As you can see here, the allosteric site is separate from the active site. However, molecules can still attach and interact with the allosteric site. So activators are molecules that bind to the allosteric site but increase the ability of the enzyme to react because it helps maintain the shape of the enzyme. And this is seen through ADP in glycolysis. On the other hand, inhibitors are molecules that bind to the allosteric site, but they reduce the activity of the enzyme by making it lose the shape of the active site. And an example is ATP in glycolysis. There are two types of inhibitors. There are competitive inhibitors and non-competitive inhibitors. Competitive inhibitors interfere with the active site of an enzyme and block off the substrate so it cannot react. As you can see here, the inhibitor takes the place of the substrate so no reaction can occur. On the other hand, non-competitive inhibitors change the shape of the enzyme by binding to the allosteric site. This prevents the substrate from attaching to the active site because the shape is now different. As you can see, the inhibitor changes the shape of the enzyme's active site, which was a V but is now a rectangle, so the reactants cannot attach. Feedback inhibition is another type of regulation for enzymes. In this case, the product of the last reaction acts as a non-competitive inhibitor to the first part of the reaction. This is a way to ensure that the products of the pathway are regulated so they're not produced unnecessarily. If we take a closer look, let's say this is the pathway and it produces this circle as the product. This can then bind to the allosteric site if there are too many, and then change the active site to momentarily pause the pathway. Enzymes are definitely designed with a purpose because they are so interconnected in everything that occurs in our bodies. Without them, our bodies would not be able to function. There are so many conditions in which they need to work, like temperature. Each enzyme has its own optimal temperature at which it works best. It's usually 37 degrees Celsius, which is the temperature of the body. The same goes for pH. Enzymes each have an optimal pH at which they work the best. And if it is not at the correct pH, the enzymes can get denatured, and this is when the hydrogen bonds will break and the properties of the enzymes will be lost. Concentration of substrates is another one. As more substrates are added, the rate of reaction will increase until all the active sites are used. Similarly, concentration of enzymes. The more enzymes, the longer the concentration rate. This is different than concentration of substrate, where there's a point of saturation. And number 5 is the effect of competitive inhibitors. Competitive inhibitors can really stop reactions from occurring by blocking substrates from getting to the active site. All of these factors are possible ways for enzymes to stop working. However, each enzyme is placed in a certain condition in which it will thrive and work at its best, and that is definitely designed with a purpose, like pepsin. Pepsin is in charge of protein digestion, and its optimal pH is 2, which is exactly where it is in the stomach, which has a pH of 2. Plus, the temperature is perfect for it, which is the body temperature. Everything about enzymes is designed with a purpose. For example, feedback inhibition shows how complex it is, or how each enzyme is specific to its own substrate. Especially the fact that enzymes remain unchanged after reactions shows how well they are made to be used again and again to maintain life. In short, enzymes are truly the most important concept in biology, because without them, we wouldn't be able to live. They speed up the rate of virtually all the chemical reactions that occur in the cells, making them possible to occur and efficient. They are in everything in our life. For example, digestion. They help our bodies break down large, complex molecules into substances that our body can use to function. For example, pepsin and trypsin are used for digestion. Another option is metabolism. Enzymes are known to speed up these metabolic reactions in order for our body to be able to function and move. Similarly, in respiration, the carbon compounds pass through a series of enzyme-catalyzed reactions in order to give our bodies ATP. For example, coenzyme A and acetyl-CoA are used before the Krebs cycle. In muscle cells, there's enzymes used like creatine phosphokinase, CPK, which allow our muscles to work, even in our nerves like beta secretase. Enzymes are especially important in DNA replication, where the enzymes unwind DNA coils and copy the information, making DNA replication and life itself possible. Overall, enzymes are essential to life. Thank you so much for listening, and I hope you have a great day.